Good day and welcome back to our channel. Today we will discuss equipment which are being used in the microbiology laboratory. We don't do a formal video where we've been do the part 1 of these equipments. Today we will do the part 2 of the equipment where they use for laboratory, especially microbiology laboratory. And it's important to know this equipment, understand these different equipment, so that if you enter laboratory to go do different experiments, you no go dey surprised or you no go see any equipment and say, I never see this one before. Now for this video, we will just take 8 of the equipments wey they use for inside microbiology laboratory, discuss these 8 equipments and wetin we dey use these 8 equipments do. The number 1 equipment wey we go discuss na equipment wey we dey call incubator. Incubator dey very important for microbiology laboratory, and the incubator e dey help create environment with the right humidity and temperature wey we fit use to grow many microbial culture. So many times when we put our microorganisms, put them on top all these are different culture media. If we do that one finish, we will come carry that culture media, wrap am well, come put am inside this incubator, because the incubator will dey fit na like box, and we fit set am for particular temperature. Many a times we dey set am for temperature of about 37 to 42 degrees, because many of the microorganisms wey we dey study for this part, or many times the microorganisms wey we dey study na microorganisms wey dey affect health of human beings, and usually body temperature na for around 37 degrees Celsius. So e dey important. Many times the incubator we dey set am for 37. So apart from temperature, also the incubator dey set humidity, how wet the inside of that environment go dey, and e dey important because e dey encourage the growth of all these microorganisms. So that one na the first equipment wey we go discuss na incubator. Now after incubator, you get another equipment wey we dey call autoclave. Different types dey, big one dey, small one dey, and this autoclave we dey use am do wetin we dey call sterilization. Now any equipment wey we wan use for inside a laboratory, many times many of our equipments dem dey made up of metal or glass. Now many of these equipment, in order to use them, we no want any microbial contamination for inside all these equipment, so we dey carry them go for inside autoclave, where we go use steam at high pressure to use to destroy anything wey be like microorganism, whether it be their spores or their cells. We will destroy them totally using that steam and high pressure. Now many a times, if we look at the temperature wey we dey use for inside autoclave, that temperature na usually around 121 degrees Celsius, so e dey very hot, and na steam and heat together. Also, as we get that very high temperature, we dey get very high pressure too. So the thing dey work with both high temperature and high pressure. The pressure wey many of our autoclaves dey work at na 103 kilopascals. This one na the unit wey we dey use measure pressure. So na very high pressure situation, both high pressure and high temperature na we dey use run the autoclave. So incubator dey, autoclave dey. The next one na biosafety cabinet. Many big laboratories dey get all these big cabinets, and these cabinets dem be cabinets wey be say the environment for within the cabinet dey controlled. We no want any kind of microbial contaminants to dey enter that cabinet. So we'll get big cabinet like that. Many big laboratories dey get them, and many at times many of these cabinets na glass they use make am, and many of them too go dey wey be say they go get places where you fit put your hand, they run all the different experiments wey you wan do within the biosafety cabinet. So this biosafety cabinet dey very important. Many big laboratories get them, and na controlled environment where we fit dey do many experiments, dey do many reactions, and everything go dey controlled. Microorganisms no go fit see their way into this biosafety cabinet. This one dey important. The next one na centrifuge. Centrifuge na equipment wey we dey use for laboratory, wey be say we dey use am spin samples, any material or sample wey we get, wey we wan separate that material or sample into its component part, meaning say the heavier ones go dey down, the lighter ones go dey up. We'll put that kind thing wey we want, that kind sample wey we wan separate, we'll put am inside centrifuge. That centrifuge go come spin the material at a very high rate, very high speed. Many times get speed wey we dey call 1000 revolutions per minute. Another one dey wey we dey call 5000 revolutions, 10,000 revolutions, depending on wetin the scientist wan do. We will spin those samples very well for inside that centrifuge. If you spin am finish, at the end of the day, wetin dey happen na say the samples wey dey heavier go settle, and the ones wey dey lighter go dey on top. So that sample go don dey divided on the basis of how heavy wey the different components wey dey inside the sample be. So that one na our centrifuge. Even for hospital, e get our brothers wey be scientists, wey be medical laboratory scientists, even doctors, they dey use wetin we dey call hematocrit centrifuge, and that one they dey use am on top, they dey use blood samples on top am. E go separate those blood samples into all the component parts wey dey for inside the blood, and they go fit tell you different values like your PCV and other important information. So this one dey important to note. Also, we get wetin we dey call incubatory shaker. Incubatory shaker na another equipment wey we dey use for lab, and just like the name bin imply, incubator and shaker. Now, e get many experiments wey we dey do for inside laboratory, and many samples wey be say as we dey run those samples, we want to dey agitate the sample, we want to dey shake the sample, we no want make the sample rest. For science, we dey call am homogenize. So we go dey, dey disturb that sample even as the sample dey for inside the incubator. So for any experiment like that, we dey usually put am inside wetin we dey call incubatory shaker. So that thing na incubator normally, then e come get some things for the base wey go dey help us dey agitate or dey shake our sample. This one dey important, and e get many experiments wey we dey do wey be say we need to dey agitate those samples. So that equipment dey, we dey call am incubatory shaker. We also get equipment wey we dey call pH meter. Many different types of pH meters dey. Some dey wey be say they go arrange them for on top table, some na handheld. But wetin pH meter dey do na say e dey help to measure pH of any solution or any sample. We fit measure the pH. So pH, remember say pH na degree of either how acid something dey, or how basic, or how neutral e be. So for any side, we know say that one na wetin we dey call pH. So we dey use the pH meter to measure the pH of any solution or any sample. Now, many times too, we fit use am to adjust pH. Let's say our pH dey for particular level and we wan reduce am, we wan make am more acidic. We go dey add acid to that solution, and we go dey use the pH meter to dey check, to adjust till e get to the level wey we want make